Hello everyone, my name is Freddy Gonzalez and today we will learn how cells produce more cells and the process of mitosis. In the time that I'm telling you this, your body will have made millions of new cells. Making new cells allows you to grow and replace cells that have died. Some cells are replaced less often, but your body is constantly making new cells. As you grow, you pass through different stages in life. Your cells also pass through different stages in the cycle that they have, which is called the cell cycle. The cell cycles begin when the cell is formed and ends when the cell divides and forms new cells. Before the cell divides, it must take a copy of the deoxyribonucleic acid, DNA, which is the hereditary material that controls a cell's activities, even the making of new cells. The DNA of a cell is organized into structures called chromosomes. But what are the chromosomes? Chromosomes are not visible in the cell's nucleus, not even under a microscope, when the cell is not divided. However, the DNA that makes up chromosomes becomes more tightly packed during the cell division and then is visible under a microscope. Most of what researchers know about chromosomes was learned by observing chromosomes during cell division. Copying chromosomes ensures that each new cell will be an exact copy of its parent cell. But how do cells make new cells? Well, it depends on which type of cells we are talking about. It could be prokaryotic with no nucleus or eukaryotic with a nucleus. Prokaryotic cells are less complex than eukaryotic cells are. Bacteria, which are prokaryotic, have ribosomes and a single circular DNA molecule, and don't have membrane-enclosed organelles. Cell division in bacteria is called binary fission. The cell on the image is undergoing binary fission. As you can see here in this image, binary fission results in two cells that each contain one copy of the circle of DNA. Look at this. Here you can see the differences between eukaryotic and prokaryotic cells. Eukaryotic cells are more complex than prokaryotic cells are. The chromosomes of eukaryotic cells contain more DNA than those of prokaryotic cells do. Different kinds of eukaryotic have different numbers of chromosomes. More complex eukaryotics do not necessarily have more chromosomes than simpler eukaryotics do. For example, fruit flies have 8 chromosomes, potatoes have 48 chromosomes, and humans have 46 chromosomes. In this figure, you can see the 46 chromosomes of a human body cell, lined up in pairs. These pairs are made of similar chromosomes known as homologous chromosomes, because they have the same sequence of genes and the same structure. Mitosis is the process by which the eukaryotic cells divide or reproduce. Mitosis ensures that each new cell receives a copy of each chromosome. In other words, it is an exact copy of the parent cell. This process, known as mitosis, is divided into 4 parts. The image that you see now shows the cell cycle and the phases of mitosis. In animal cells, mitosis has 4 parts, and they are prophase, metaphase, anaphase, and telophase. There is a previous phase called interphase, the one that takes place before the mitosis begins and in which the chromosomes are copied. The first phase of mitosis is called prophase. At this phase, the chromosomes condense from long strands into rod-like structures. During the second phase of mitosis, called metaphase, the nuclear membrane is dissolved and the paired chromatids aligned at the cell's equator. During the third phase of the mitosis, the chromatids separate and move to opposite sides on the cell. After complete separation of the chromosomes and their extraction to the spindle poles, the nuclear membrane begins to form around each group of chromosomes at the opposite ends of the parent cell. The nucleoli also reappears in what will eventually become the two new daughter cell nuclei. When telophase is complete and the new cell membrane is being formed, the nuclei have almost matured for the pre-mitotic state. The final step is telophase, involves the initiation of plasma membrane cleavage between each of the new daughter cells to ultimately yield two separate cells during the next phase of cell division, known as cytokinesis. There is another phase that takes place after mitosis concludes. It is cytokinesis. In cells that lack a cell wall, the cell pinches in two. In cells that have a cell wall, a cell plate formed between the two new cells. Well, this is all for today. I hope you enjoyed this video. Thank you very much.